Thank you everyone for joining the presentation, for attending, and please feel free, as Tudor said, if you don't understand something, please interrupt and I'll try to clarify right away. Okay, so the title of today's presentation is Ontogeny and Syntax of a Memory Index. And just to introduce the topic, my main interest is in studying internally generated representations, and these are defined as mental representations of physical objects or events that are not currently present. So some of them, most famously, have been studied as a form of memory, right? They deal with our past. Some are engaging with our future, such as imagining, planning, a cognitive map where you can take detours and shortcuts through a map. It could be a spatial map, it could be a cognitive map. So this is a key term in the type of research that I do. Probably the most genuine types of internally generated representations occur during sleep, particularly during dreaming, when the brain is fairly disconnected from the external world. And at times they can take an abnormal form, the internally generated representations, such as the case of hallucination and delusions. So this interplay between past and future and the role of experience in generating internally generated representations has been a fascinating topic, and starting with philosophical debates from, say, British empiricists who have argued that experience is absolutely required to form any representation, probably because at birth our mind has no innate ideas. It's a blank tabula rasa. And then rationalists Leibniz, Kant, and many others have argued that, in fact, we are born with some concepts a priori, such as at least space-time, I guess some of the math concepts, some of geometry in general beyond space, are not derived from experience, but rather as preconditions. So later on, I will touch on this type of debates and how they enter the field of neuroscience. But here I would like to say that a brain area called hippocampus has been intimately related to all these four aspects that I listed, four aspects of internally generated representations. I don't know if you see my mouse. Can you see it? Okay. Starting with the memory, this is the famous case of HM. Scoville and Miller published in 1957. An account on this patient that lost ability to form new memories. So this is a form of anterograde amnesia following bilateral hippocampal lesion. It took 50 years, from 57 to 2007, to realize, for the community to realize that, in fact, the same patients, the same subjects that lost ability to form new memories also have trouble imagining and planning. And there were a couple of papers, a couple of groups published studies in the same year, 2007, patients with hippocampal amnesia cannot imagine new experiences. Even later, it was realized that the same patients have trouble dreaming, and that is not due to their lack of memory, because their short-term memory is good. So when they are woken up after an episode that could have contain a dream, they cannot remember the contents, or they cannot dream about it. And finally, there have been a number of studies. This is a review summarizing some of the relationships between hippocampal formation and schizophrenia. In fact, I would like to go back to this study here in 1957. There was a list of subjects, there was a list of patients that had bilateral hippocampal lesions. HM was just one of them, but all of the others had psychosis, schizophrenia or bipolar. And in most cases, after hippocampal bilateral lesion, the positive symptoms of psychosis have diminished significantly. So this seems like a brain area that could be considered as a generator, internal generator of prerequisites for mental representations. So the anatomy of hippocampal formation, starting with the human brain, hippocampus is a medial temporal lobe area. It's a C shape located as shown in this figure. In the rodent, has similar shape, but occupies a much larger proportion of the whole brain. And if we perform a cut around this level where the colors are, we can reveal the intrinsic circuitry of the hippocampal formation with the classic trisynaptic circuitry, entorhinal cortex to dentate gyrus, then to CA3 and then CA1, and then back to entorhinal cortex, which has been the attention of a lot of research groups. Yeah. Speaking in terms of physiology, I'm going to skip through LFP studies, just to mention briefly that indeed Yuri Buzhaki in 1989 proposed the two-stage model of memory formation. Memory is generally understood as having two stages, encoding and consolidation. And encoding is believed to occur, as shown here, while the animals explore, in this case, a linear track. This is contributed by external stimuli, so this is by all means experience-driven. And here is a sequence of 8 place cells. These are cells that fire along the trajectory of the animals at specific locations. So they have a tuning curve that is tuned to specific locations along the trajectory. And this takes about 4 seconds for the animal. So this fields sort of cover this area around this amount of time. When the animal goes to sleep, in particular the non-REM sleep, there is a new pattern takes over, and some of the most salient features are the large irregular activities, which when filtered in the 150 to 200 hertz reveal high frequency oscillation called ripple. So you have theta oscillations, you have ripple oscillations. And ripples have been at the forefront of brain mechanism underlying memory consolidation. So there have been hypotheses, and more causal experiments are being performed to sort of indicate that what happened during ripples may be important. And what happens is, at times, a replay of the previous activity. So this, look at the time scale here, it's about 20 times compressed, but what is preserved is the order in which these neurons were activated. They are reactivated or replayed in the same order in a significant number of occasions. Not the whole sleep that follows is dominated by this pattern. There are other patterns that back then in 2002 were not understood. So this temporal compression was believed to be something specific to the non-REM sleep that will then facilitate memory consolidation that would not be able to happen while the animal is awake. However, a few years later, just for about 4 years later, we showed that relatively the same type of temporal compression of sequential activity, say as it happens here, can also occur within theta cycles of oscillation even during the exploration. So each of these place cells will be mapped by a theta oscillation that's underlying, and within each theta cycle, I'm talking about place cells that overlap, say around here, the order in which these neurons fire will be preserved and will be compressed about 15-20 times. As shown here, this is a cartoon. This is showing essentially, it's a square linear maze mapped onto a theta cycles, as you can see. So they're highly compressed, and then one arm of the maze is mapped within the theta cycle, just like it happens during sleep. This is another illustration of this. This is a theta sequences that occur within theta cycles. And what is interesting is that the distance, look here or look here, doesn't matter, it's a cartoon. The distance between any two place cells nearby and far apart, which is shown here on the x-axis, is mapped at 2 time scales. One is at the so-called behavioral time scales. If you divide the distance between any two place cells by the relative speed of the animal, average speed of the animal, you get a temporal bias between activation of these neurons. So this is shown here. So these are called place cell sequences. So this is the time scale at which they are activated in time, y-axis versus in space. At the same time, we can keep the x-axis identical, and then we can see that at about 1 order of magnitude faster, the order of neuronal activation is preserved within theta cycles. So this is many ways of showing the exact same thing, that there is a temporal compression of neural activation as the animal is exploring this environment, which is then replayed in the sleep after at about the same or slightly faster compression. And probably more intriguing is a discovery published in 2011, by which the de novo run and de novo placeholder sequence is established on a linear track, and these are adult naive animals that were kept in their home cages lifetime for their whole life, except for surgery and such things where they were not conscious. And then on a particular day, once electrode locations were stabilized in the CA1 area, dorsal CA1 area of the rat hippocampus, sleep activity was recorded, and the animals were allowed to run on a linear track. And then we looked back and found that the same neuron that fired in this particular order during the run had already fired in this order in a significant number of instances during the sleep before this time, not after, and also after. So we call this phenomenon pre-play. Because you can see here, neurons like to have multiple fields, in particular in this type of high novelty environments. Later on, they tune up more. We employed another analysis tool, which is called Bayesian decoding. And without getting into too much details, this is an analysis that is using all the activity. So all the multiple peaks here, it's getting a probabilistic order of neuron firing and bin by bin position of the animal as decoded as encoded by this number of neurons. And then that is introduced into a Bayesian decoder, which then predicts the position of the animal based on the instantaneous activity of ensembles of cells, with the prior being this average map, right? And in yellow, I'm showing the actual position of the animal. So the space is on the y-axis, time is on the x. So as you can see here, the animal is sitting at the end, resting at the end of the track, one end of the tracks. And you can see the position is relatively stationary. And the decoder, which is this heat map, decodes the animal with low error. And once the animal starts moving, it's moving from one end to the other, time goes up also. The decoder follows the animal position. And when the animal is resting again at the other end, the decoder is fairly accurate. So after building this decoder, which then maps or reconstructs the position of the animal during run based on activity during run. So it's just more of a technical establishment of this decoder. We then use this decoder to decode activity or potential position of the animal based on activity of the same neurons in the sleep prior to this experience. So here the animal has not yet seen the linear track, but what we can see at times, the decoder establishes or depicts virtual trajectories through a space that was only later explored. And this is not all sleep, and not all events are like that. These are selected, but there's a significant number of them that are correlated with the future trajectory of the animal. So this is to say that the network, in fact, is not assembled on the spot as the animal runs, or the activity is not purely instructed by that experience, but there is something pre-configured in the network by which neurons form cell assemblies, form sequences that cannot be escaped at the time of encoding any future novel information. So the building blocks of neural organization are being used, and this is probably what we're seeing here. When the same animals were put to run on 3 tracks, so run on track 1 is depicted in red, blue is another track, track 2, and then orange, track 3. We can see sequences that are unique for each track, and then they all have, this is just one example, but they all have significant amount of preplay prior to. This is another. By the way, the cell IDs here of the neurons are unique. So you can see that the order of neuronal activation on different tracks is different, yet we find preconfiguration for all 3. And then when we look at all significant events for the 3 tracks and plotted them in a 3D space based on the correlation value with each of the 3 tracks, we see that they tend to cluster with little overlap in some spaces whereby preplay for one track is more alike between the events, of course, grouped by the correlation with the same future track. So the idea is that there is a repertoire pre-configured temporal sequences that can be later be used to encode multiple novel experiences. So this is a brief history of models of spatial temporal sequence expression in the area CA1. The dominant view up until, say, 2010, 2011 was the instructionist view, in which during the experience, somehow external stimuli drive, in conjunction with internal ones, I guess, but drive the sequential organization of neurons, such as shown here, A, B, C, D, E, to map a place cell sequence on this track. When looked experimentally, when looked before, no such sequence could be observed. And when looked in the sleep after, there was a strong replay of this activity, temporally compressed. As I shown before, there were no other sequences that were at least reported to exist. So in our work in 2011, we demonstrated that these sequences are in fact internally generated. The network is pre-configured rather than blank slate or tabula rasa, in that we find the same sequential activation of animal runs on a linear track, just as they found before. But looking back in time, we did find sequences correlated with the one on the track in the temporal domain during sleep. There were other sequences there too, and there is replay of this activity, but there are always other sequences. Now, the animal being exposed to only one track, we didn't know. We didn't have a template to correlate with. We didn't know exactly what else is going on in the network. So when we exposed the animals to 3 separate tracks, we found that in fact there are clusters of sequences that will be correlated with either of the 3 tracks and with very little overlap. So I think of this as a selectionist view. The sequences are selected online from a pre-existing repertoire and modified and made somehow specific to that particular experience, which later can be replayed and potentially gain meaning when it comes to association to a particular experience. And shown here in a thicker line is that replay is a stronger depictor, a stronger representation of a particular experience compared to preplay. Whether you consider strength of correlations, whether you consider of significant correlation, or incidence of significant correlations. So this is the working model that it's currently functioning in my laboratory, and I guess beyond that in the community. We have the phenomenon of process of encoding, in which pre-configured sequences of firing are selected during novel experiences and gain new representational meaning via associations with novel multisensory external stimuli, which are not particularly investigated directly here. And then second phase of consolidation is initiated via plasticity in the replay of new firing sequences during subsequent rest and sleep epochs. So there's a process by which that particular sequence that was experienced is somehow better represented now in the sleep after than before. And finally, as more of a general view, compressed temporal sequence of firing could represent an index code for the spatial temporal order of events in a memory episode. I'm seeing this is a code because not all details in the perceptual world of the experience are encoded in the hippocampus, but they're somehow indexed there. So brief recap of what I've shown so far. We have place cell sequences, and at the same time, we have decoding or reconstruction of animal position based on the activity of these neurons at the instantaneous time scale. Then we have the phenomenon of preplay, which precedes and somehow preconfigures the network, which then facilitates rapid selection and modification of the sequences online. We have data sequences that bind recent past with current and immediate future. Those sequences are within the time scale of plasticity within data cycles. And finally, we have the phenomenon of replay, by which the activity that was, the sequential activity that was expressed during a particular experience is reactivated, and that replay would be stronger than the preplay. So I envision these two stages, encoding and consolidation, of including replay and selection of replayed and data sequences, and then plasticity and replay as the initiator of memory consolidation. So this data were obtained essentially worldwide in the adult, and there was essentially a gap in neuroscience regarding of their developmental timeline. So in particular, triggered by the fact that preplay exists in naive adult animals, this particular fact drove my interest into finding out when are they emerging and what are some of the conditions and determinants of this developmental timeline. So here we investigated that in postnatal third and fourth week postnatal life of rodents. Just a quick orientation that the eyes are opening at postnatal day 14. They're born with eyes closed. They're essentially blind until the end of their second postnatal week. And then place cells have been, there were a couple of back-to-back papers. This is from the O'Keefe group, this is from the Moses group, showing that the earliest age in which you have a adult-like maturation of place fields, meaning tuning to a particular location rather than firing everywhere, happens at postnatal day 16. So we knew that. And then grid cells, another interesting pattern of activity in the middle temporal lobe related to space. They mature around postnatal day 21, 22, so later than CA1. And then another phenomenon which exists in humans, and there is a model for it in the rodent, which is infantile amnesia, also known as the normal forgetting of events that occurred in our early part of life, say before we were the age of 3 to 4, which are not remembered in adulthood. So there is a age at which is believed that in rodents infantile amnesia ends and sort of ability of the animal to form long-lasting memories emerges, which is postnatal day 23, 24. So we decided to perform our recordings in naive animals of different ages, starting with postnatal day 15. So they have some access to visual input, even though it's not very mature. And then we can capture maturation of place cells, grid cells, and the end of infantile amnesia. So how did we do this? We implanted rats at different ages, and essentially we stabilized the electrodes within the dorsal CA1 area, which is deeper than the neocortex. So it takes time to stabilize the electrodes there. So it took about 3 days. So for P15 recordings, P15 rat recordings, the implants are at P12 and so on. So we implanted from P12 to P21. And we implanted with these types of silicone probes, either 4-shank or 6-shank each, to a total of 64 channel, 128 recording channels. And here is a sample of, is an image, a histological image of the 4 shanks reaching the CA1 area and recording simultaneously neuronal activity. So one on the left and one on the right hippocampus. So probes are lowered over the 3 days, stabilized into CA1 area of the hippocampus, and then once stable activity is obtained, the typical experiment that we do, which is sleep pre-run in naive state, is recorded. Then the animals perform a de novo run, several laps, 20, 30 laps, which is called run 1. And then we have post-run sleep, and at times we have another run session. This is an example of one of the better recordings in which every single shank collected a good amount of neurons. And nowadays we can exceed 100 neurons per recording. This is to show something that we were unsure about, if the animals will actually run on this or explore this linear track. And you can see this is a postnatal day 15. This is the youngest they were used, which is stationary at the track end when we placed him there, but then starts exploring, then stationary, and then keeps going, and essentially does a lot of full laps, probably trying to escape and not finding, and then this way mapping this linear track. I'm not going to go over all the details of this experiment to leave some room and time to explain some of the newer experiments. But basically what we found is a three-stage development of compressed temporal sequences, and it will hopefully become clear why we call these stages. Initially, at postnatal day 15 and 16, we observe representation of individual locations. We call them stationary frames. So what's shown here is this is space, this is time. This is a collection of at least 5 neurons that were recorded together and fired in this sleep frame. And they map an individual location. They do not have the sequential type of activation that we see later in adults. In what we call stage 2, we have a representation of increasingly longer trajectories. So starting with postnatal day 17 and ending at postnatal day 22, there is a increasingly higher and higher slope in this depiction of the experience towards a full track. So we have this gradual increase. We have a network preconfiguration. This is in sleep before, also in the sleep after. Data sequences are not yet observed across all these ages. And we also do not observe any plasticity in replay, meaning the experience did not seem to change in a detectable manner by us. The sequential organization of neural activity and the way the network depicted that particular experience after compared to before. And in stage 3, which we name because theta sequences emerge around that at that age, and then plasticity in replay emerges also in conjunction with theta sequences. And then activity looks more or less adult-like at that stage. I'm not saying everything is adult-like, but these particular features that we looked at, and here is an example of theta sequence expression, and then an event could be replay, could be preplay, but here is the shift in the so-called plasticity in replay, by which replay expresses stronger than preplay the experience. And this, interestingly, coincides with what other groups have reported, the emergence of episodic-like long-term memory, or the end of infantile amnesia. So this is a good time to sort of speculate or indicate that this could be one mechanism by which memories that are formed, and we know they are formed even in stage 2, memories that are formed at those ages can last long term. Okay, so again, we were intrigued by the emergence of all these patterns in the absence of any experience, right? So this postnatal day 23, 24 rats were naive, also kept naive, and then they expressed a preplay, theta sequences, and plasticity replay upon a single experience, multiple labs, but a single session. So we wanted to understand better if probably the activity in the home cage, right, for the 24 days or so, 23 days or so, played any role. So this is not an easy experiment to perform. You know, without stressing the animals, we aimed to somehow deprive them of a particular spatial experience that we thought could have been instrumental in creating in the first place this preconfigured network. So upon a closer look of how they live in the so-called cuboid cages, which have this shape, they have exposure to linear borders, which are probably not that short given the size of their body. They have exposure to planar floor. They have a distinct vertical boundaries. They have distinct start, middle, and end of a trajectory, and they have 90 degrees corners, whether they are horizontal or vertical. So we decided to deprive them of experience with what could be encapsulated as Euclidean geometry, right? By rearing them from birth inside non-Euclidean curved space. And this is one form of non-Euclidean curved space. There are other ones, but this is a convenient one. This is a translucent sphere. It's made of 2 hemispheres that are sold as light domes, basically. For big halls, you have those protective, bulb protecting or diffusing hemispheres. So here we have curved borders We have curved floor. We have pretty much similar start, middle, and end. There is no mark. There is no landmark. There's no boundary, and there are no corners. So we don't know which particular features would be the key one, but we decided, at least for the first pass, to basically deprive the animals of all and see if we get anything. And just to mention that this is a view, camera view of the inside of the sphere. This is the bedding where they are housed together with the mother. It's just to say that they're not restricting their activity and movement to the bedded area. So they do explore and they do move. In fact, in our measurements, they move not less than, not more, not significantly more than the cuboid raised animals. All right, so what do we find? We have exact same protocol with the difference that the animals are sleeping all the time in the environments of their sort of denomination. So cuboid stays cuboid and sphere stays sphere. And the only experience that the animals had, these are postnatal day 24 animals, in their lifetime with the linearity was running on a linear track for a full session of say 20 to 30 minutes. Then they go back into sleep in the same cages and then again run. So what we observed in the place cells, and this will be the sphere ones, are a little bit detuned, but generally they can establish a pretty understandable, pretty distinct sequence of activation along the trajectory of the animals. So we know that that particular experience in Euclidean space is not required to generate relatively well-tuned place cells. When we perform decoding of animal position and reconstruction of animal position from the activity of place cell ensemble as the animals were running, we could see that in fact the error rates were about the same. That's shown, quantified here. The ensemble of place cells of sphere-reared animals were able to, when analyzed, were able to reconstruct the position of the animal as well as the cuboid. And then both groups were much better than shuffle. We scrambled the bins of and the correspondence with animal position. But data sequences were also present at P24 in cuboid. This is when they emerge in cuboid, and they were already present in the sphere. There didn't seem to be any significant difference between the quadrant ratio, basically the method that we use to assess data sequences. When we looked at the activity, on the analyzed activity during sleep in the sphere-reared animals, we could see clear examples, significant number of clear examples of preplay and replay activity. We quantified it here. Preplay is above chance, replay is above chance, and there is also a stronger replay than preplay with regards to that particular experience. But they were lower in incidence compared to the cuboid, though present. The probably equally interesting, or potentially more interesting, a kind of a positive finding, is when we put the animals to run on day 2 on a U-shape, a 3-track maze. Basically, they were sleeping in their cages. They were rerunning track 1, which is now familiar, and then access to tracks 2 and 3, which was previously blocked by 2 barriers, has been granted, and then now the animals run the U-shape. And we have data to argue that this segmentation here by the 2 barriers made these 3 tracks appear as different, plus the head direction. So what we found is when we looked at significance of preplay as far as its specificity to a track. So a preplay event, a frame in sleep could be significant for one track, but could also be significant with another one. When we impose the rule that only events that are significant for one track and only one track should be included, then in the cuboid, we still had a significant proportion of events being uniquely depicting tracks, a little bit more depicting some overlap. but in the sphere animals, that dropped to chance. That specificity dropped to chance. At the same time, we looked at the activity of place cells in the cuboid and sphere animals across the 3 tracks, and we could see that neuronal activity in the sphere-reared animals, this day 2, postnatal day 25, is less specific to one track. They like to, or they are activated on multiple tracks, sometimes around the same location, sometimes not. Whereas the cuboid-reared animals, in the cuboid-reared animals, the cells are either specific to one track and don't fire at all in the other, or fire in very different locations. So we computed at the population level a correlation between the ensemble of place cells across the 3 tracks. And we also looked in the development. So we had the data that we had collected for all ages. If you'd like to focus on sphere versus cuboid, we found a difference. So the population vector correlations were higher. Higher means less specific to each track. They were represented more similarly in the sphere versus cuboid. And looking down into development, this is something we observe. P16 animals, which is day 2 of the P15, it's their ability to discriminate, to parent separate, if you'd like, across the 3 tracks was the worst. And then the sphere animals were somewhere around postnatal day 17, 20. So we found that, in fact, not having exposure to multiple, not being able to create a repertoire of multiple sequences based on experience in the cages did not prevent the emergence of preconfiguration, but affected the repertoire, affected the extent to which later on experiences could be discriminated and separated as distinct. So I'd like to think in terms of network competence and network performance. So network competence to be activated in compressed temporal sequences that will later be selected and match activity during a new experience, and then later will replay that activity, is not affected, it's still there, the competence is there, but the performance by which they do it would be affected, and it would require some specific experience. So we continue to expose animals over the next 2 days. So with day 3 and then day 4 of the experiment, again, they were sleeping in their cages, and overnight they will also be sleeping in those cages of corresponding geometry. So we know exactly how much experience they had on linear tracks. Again, they were running on track 1, and then on a novel track 4, and then on a novel track 5, and then repeated track 4 and 5 with some slip in between. On day 4, track 5 became the baseline, and then track 6 was another novel one, and so on. So after 4 days, the animals were exposed to a total of 6 different tracks. And what we observed, this is day 3 here, this is day 4. By day 4, basically, the specific preplay was above chance, and let's see, it was not different from the one in the cuboid, and pretty much there was no difference between the groups, with some observation that happened already by day 3, but it was stronger in day 4. This is during sleep, prior and after. Also, parent separation during the run was the same in the cuboid and sphere animals, and it was actually the same as the day 2 of cuboid animals. So we see that by end of day 4 or during day 4, all of these patterns that we've detected as deficient or deficits in sphere-reared animals have recovered. So with this, I'd like to conclude this part by saying that experience with Euclidean geometry is dispensable for the emergence of place cells and of preconfigured and plastic temporally compressed sequential patterns. And we think of this as the nature aspect driving this activity. That pattern separation between multiple linear contexts depends on prior experience in Euclidean space, and we can think of this as nurture, there's experience. And that experience with Euclidean geometry over 3 to 4 days at P24, 27 is sufficient to counter the deficits induced by deprivation from birth. And we can think of this as something like a niche for which the animal is tuned to live in. This is a cartoon type of compression of this type of information. We did a bit more analysis that's available right now in a bioarchive paper, hopefully soon in a published one, in which we found that there is a repertoire of neural sequences that cluster into multiple clusters in the sleep before in the cuboid animals, but only in fewer clusters. So there's less distinctiveness in the network when it comes to future experiences. All right, let's see. Okay, we got a bit more time. So again, back to sequence coding in the hippocampus. Given that the network, as expressed during sleep, seems to constrain the patterns of activity as they will happen in the future, we were asking whether this can be used as a predictive code rather than just being a correlate of what happened during RAN between what happened during RAN and what happened prior to RAN. We were asking, moving forward from the sleep network activity, if we can predict to some degree the order in which the identity and the order in which the neuron fire in any novel environment, and what would be in that case the underlying neural syntax, as called. So what we knew at the time was that that two I guess competing or dual perspectives on sequence coding in the hippocampus. At one end was the externally driven. I know by that time we were already shown that they are not only externally driven, but just for the sake of placing our neural findings into the context, we have this sort of extreme view in which there is no pre-configuration that could be used during the run. And these are, each circle is a cell, this will be a network, this is a sequence as the animal runs. There are, say, 3 tracks, there are 3 sequences, and when look back in time, in the sleep session, we cannot find any correlate, right? So this is it indicating that the network has very high capacity to encode new locations, new sequences, because there is no constraint. But it will be very low efficiency because probably the animal will have to revisit in the exact same manner the same locations in order to form a memory. So often we want to repeat an experience in order to remember it well. At the other end would be what we have described as internally generated extended sequences. And this is an extreme view in which the sequence as the animal or as the network has experienced during RAN was identical with no modification from one that could have existed during sleep. This will probably be associated with a very low capacity of the network because you will have to have you can only have experiences that match the number of such sequences that exist, but it will be highly efficient when it comes to memory because it will be just a reminder of something that essentially has already been there. So we were wondering whether there's something in between that could be intermediate level of maybe not very high, but good capacity, and maybe not very high efficiency, but good efficiency, the middle ground. So in order to look for this, we had essentially similar data sets, sleep, run, sleep. This is adult now, and then could be multiple runs and multiple sleeps, but on the same track for now. And we would look in these frames of activity as described for any potential sequential activation of pairs of neurons this time. They would repeat more than by chance. Okay, they would repeat across frames. And here is an example of, there's 76 cells, but shown here are a few. These 2 cells in red, the top cell seems to precede, time moves left to right, seems to precede the bottom cell in frame 1, 2, and 4, sorry. In frame 5, only one of the neurons fire, and in frame 3, the order is reversed. So this is a probabilistic order preferred order of neuronal firing that we could detect for all pairs in the network. So there are several ways to compute the state order dependency, meaning how many cells do we think, say this bottom neuron here, we looked here at one neuron before, but can we look at another neuron before? Do we have a triplet? Do we have 4 neurons that we can look at? So what is the length of that state order dependency? We found that using different methods, many different sleep sessions, to be around 1.5. In other words, one neuron activity would depend on 1, maybe 2 neurons firing before it. After that, the strength of this sequential activity is heavily reduced to a point that is not predictive. So without getting into many details, we build a transition matrix in which the probability of one neuron preceding another one is included, and that's for all cell pairs. And then we could use those probabilities, say neuron 1 precedes neuron 2 in 60% of the case, a probability of 0.6, neuron 2 predicts neuron 3, in 40% of the cases 0.4, then the probability of A, B, C would be multiplication of the 2, 0.6 times 0.4. And we did that for, we computed the predictability of sleep triplets from sleep pairs, and we find, not so surprisingly, we find that the sleep sequences at 3-level neurons were highly predictable by activity during sleep. So again, we built sort of a predictor from this, and we tested it that it worked on sleep based on sleep activity. Then we moved to the real question, which is, given that we know the sequence of place cells in the run, we asked to which degree the sequence was predicted out of 1 million possible scrambled sequences that we could produce by multiplying those pairwise probabilities of neuronal order that we computed pairwise for all pairs in the network. So this is what we did. We computed 1 million probabilities, and we added the two directions of running the 2 real sequences, and we found out that the probability of the real sequence was somewhere at the high end of the distribution of 1 million possible probabilities calculated from pairwise probabilities of all cells in the network. And here is a summary. We had 6 animals in 2 directions. And then what we found, the pink area is more than 95th percentile, yellow area is more than 90th percentile of this distribution. We find that all animals at least one direction sort of predicted at the 95th percentile or above, more so if we lower it to 90%. And then some are obviously not. When we included all the runs, as I said, the animal ran multiple times and multiple tracks at some point, and all tried to be predicted from the very first sleep, we find that those percentiles sort of organize around the high end. And this dotted line would be a theoretical chance level. If this will be no predictability in the network, it should be kind of spread out throughout 0 or so to 100. In fact, probably much more on lower. So we think this is a highly significant phenomenon, which then incited us or indicated to us the next step would be to look for these potential triplets. So we went on during the sleep activity, and first of all, we did a truncation experiment. So we had the whole sequence of place cells, which then we chopped into 2, and then we kept the order in each half, but then we reversed the order of activation of those 2 halves, and then found that this was done by number of neurons, or we can do by halves, as I said. We did by number of neurons, say, we get pretty much the same predictability from sleep. Then we chopped it in what would be 3, 4 portions in which we kept the order within each segment, but then we scrambled the order of the segment. So we did this over and over until we find that a minimum chunk that needs to be preserved as original is 2 to 4 neurons. And if we break that, then the predictability of neuronal sequence during run from sleep is affected, and it's affected more and more the smaller and smaller the chunk is. So this was an indirect indication that this type of organization into more rigid, if you like, 2 to 4 neuronal tuplets, we can call it triplets, 3 plus minus 1, seems to be required for that predictability and seems to be happening in the network. This is simply computing the difference. Sometimes it's easier to see the difference to the dotted line, to the original predictability percentiles, and you can see a significant drop. Then we looked in sleep for the existence of such repeating doublets, triplets, and 4-cell motifs, and we found that they all are significant. More than 4 was never significant. They can always have a sequence of 5 neurons or 6 or 7, but they will not repeat that way. And then triplet seems to be the highest incidence of repeats. Then we looked at, so this is all in time, right? This is in the sleep session prior to inexperienced and naive state. Then we looked how this temporal sequence is now decompressed, if you'd like, as place cell sequence when the animal is running on a linear track. And what we find is that the duration of the sequence motif in sleep is correlated with the spatial span on the track when they become place cells. And this is for doublets, triplets, and 4-cell. So this is an indication that the network is pre-configured in what we would like to think as word-like in the description of a sequence being like a sentence, right? So you have single neurons, could be envisioned as a letter. These tuplets would be the word-like organization, and then the extended sequences would be the sentences. So here we are showing this internal tuplet motif, that this is the extended sequence, but we could retrieve these building blocks from which this was rapidly, I guess, multiplexed and concatenated during the experience. And we think this, and we calculated that this will ensure high capacity and high efficiency at the same time. So in conclusion here, hippocampal network generates predictive codes. Then the neural tuplets, triplets, are hypothesized to represent the building blocks of hippocampal network organization in temporal sequences, and preconfigured sequential patterns are used to encode spatial information. They're not just there, in other words, they are being employed. And here is a recent perspective that I put together. It has multiple aspects, but this is to show that this could be a unified or universal, if you'd like, hierarchical generative grammar encoding that could be valid across multiple biological fields. So genetics, neurobiology, and linguistics, in which, as I anticipated, neurons can be envisioned as letters or nucleotides. Then this triplet organization could be short words or morphemes, or codons in the genetic field, and then these form longer sequences that can be envisioned as sentences, or when translated, leading to peptides and amino acids. So this could be more of a universal way of organizing information and coding. And with this, I'd like to thank you and thank, of course, the lab that contributed to all this. The development was spearheaded by Usman, and adult data collected by Jeremy, analyzed by Cafe and some of the people in the lab. And of course, the funding. Okay, so if we have time for more questions, I'll be happy to.
Absolutely. This is the fun part. Thank you very much, George. And the floor is open for questions.
I have one question. Can you hear me?
Sure.
Yeah, thank you for a nice presentation, always inspiring. I was curious about this theory that is mostly hippocampal centric. How would this translate for more or less hippocampal dependent forms of memory, such as motor learning, which are less allocentric? So the information somehow is structured more on the encocentric sequences of information. And I don't know if you have any take on this translation of the model. I'd be happy to hear.
So in striatum there is a type of organization, in particular in the singing bird system, which is an analog. It's a motor system to produce. It's not a language system per se. It's a motor system to produce vocalization. There have been described syllables, and then there are neuronal correlates to that. There are sequences. Then visual cortex. There are some reports of tuplet-like organization of neuronal activity in again triplets. So this is not. I think this is not restricted to hippocampus. My laboratory has so far only looked in CA1 for these activities. We don't even know if CA3 has it quite in the same way, and other middle temporal lobe areas that are involved in memory. But there are indications that at least for the motor system and other perceptual areas, auditory system has those sequences. Even those that seem to be predictive and pre-configured to map the incoming sounds. So I think it's more of a brain type organization, but of course there could be specialization in different brain areas by which their grammar, if you like, may be different or slightly modified from this. But the concept of having a hierarchical organization probably will stand.
Interesting, thank you.
Professor.
I can add another question, if I may. It's from different. So I found it interesting that the experiments with the spheroids and the cuboids, and I was thinking, so do you think the rearing is the only element that was interfering with the space, or could be also the representation of space? So since their space was mostly Euclidean, maybe the representation itself, or what do you think?
Well,
Seminarul.
yes, so we looked at a number of parameters. First was the amount of movement, because if the animals would be immobile, you know, one could imagine an extreme case they will all sit at the bottom of the sphere, which is not the case, because sphere is 23-inch, that's what, 60 centimeter diameter. They're very big, and I could show in that photo, they do move a lot beyond the bedding area. Another factor that we consider was the amount of stress. So we measured the stress levels, and those were not different. Now, when it comes to what exactly did we do, I think I listed those 4 factors. I can add the lack of landmarks, which sometimes are useful to orient and form maps. But the animals do move, and the neurons do get activated in a potential sequence, right? It is just, it's likely that we have some additional data, which is shown in the bioarchive. We see some neuronal activation that is more similar at the ends of the linear track, as if there is an additional pattern separation deficit between the ends, as if this concept of end was new, such as the concept of multiple linear tracks rather than just one linear. So linear probably versus spherical was immediately detected, and mapped such a way, but the distinction between those was not. So I guess your question is what exactly is causing the preservation of some phenomena and then the deficit in some other. I suspect some innate factors are at play, or instruction of the network based on different modality. You know, olfaction could trigger, in adult, could trigger sequential activation of neurons and so on. But what we can say for sure, this is not in response, this is not due to experience with Euclidean geometry, as described here. So at least we can say that is not required for the generation of preconfigured patterns in the hippocampus, but it is required, and that is the nurture part, for having them in a discriminative manner. And another interesting thing is that they recover after 4 days.
Mm-hmm. I remember there were some studies, the name Elduvel comes to my mind. I don't know if it's Dupre group, that they have this 3D set up to measure the place cells and the place field formation in 3D rather than 2D. And I was thinking if the upbringing of these pups is in this 3D environment mostly. I mean, if you could imagine some tracks that follow the spheroid shape and see.
So you would like to enrich the environment rather than deprive. Is that the reason to raise them in?
Yeah, not necessarily, but the change. So to change the way you measure the place fields. So instead of looking at this linear track, to look at the track that goes
in 3D.
I don't know. Yeah, in 3D and curved. I don't know. It's just an idea that might be something interesting out there.
I think, you know, the 3 tracks already indicated a deficit. I think if you would put them on 3D kind of, you'll still see a deficit. Now 3D has its own components. They are mostly crawling. They're not really running. The speed is an issue. It could be other aspects. Vestibular system plays in. Sure, we'd like to see this, you know, if we deprive till adulthood, if recovery is slower or not at all. You know, there are a number of questions that could be followed up.
Thank you. Anyway, the model is quite interesting, and I think it will inspire people to look for it. Oh, thank you.
Yeah. Yes, please. I cannot hear for some reason.
Domnule Flonta, nu vă auzim.
Nu.
Nu se aude. Și nu sunteți pe mute. Deci, teoretic, ar trebui să se audă. Dacă puneți în chat întrebarea și o reproduc eu, dacă nu reușiți să aprindeți microfonul.